Hi, I am Chandler, Teny, and Alden and I will be presenting on the experiment of fused dimerization increases expression, solubility, and activity of bacterial dehydratase enzymes. The main focus of this paper is the manipulation of two dehydratase enzymes, Fab-A and Fab-Z, in order to increase the production of fatty acids in bacterial cultures to make a more sustainable generation of biofuels and biomaterials. There are distinct characteristic differences between these two enzymes. For example, Fab-A is a soluble protein and is relatively easy to express and purify from cultured E. coli. On the other hand, Fab-Z has been shown to be mostly insoluble and only found to be partially active. Another characteristic worth noting is that Fab-Z will dehydrate acyl intermediates of varying lengths, while Fab-A demonstrates specificity towards acyl chains that are at least 10 carbons long. In an effort to increase the production and activity of both dehydratase enzymes, the researchers of this experiment designed a construct that consists of two identical subunits of both Fab-A and Fab-Z being fused with a naturally occurring peptide linker. This will ultimately force their dimerization.
Next, we're going to go over the materials and methods used by the researchers in this article. They obtained both the Fab-A and Fab-Z dehydratase enzymes genes from E. coli, and they obtained the PFA-A gene from P. fripundum in order to create the peptide linker. Now, the two enzymes were dimerized using the linker, which corresponds to amino acids 1511 to 1538 of the PFA-A gene. And the primers used in the PCR amplification of Fab-A and Fab-Z contain overhangs complementary to either the 5' or 3' terminus of the linker to allow for dimerization. Next, the fused dimers were constructed using overlap PCR, and all PCRs were run using PFU Ultra to fusion HS DNA polymerase. The linker region was amplified using plasmid DNA from P. profundum containing the ACP domains, mixed with primers 9 and 10. Fab-A was amplified using DNA from E. coli, mixed with primers 1 and 4, and Fab-Z was amplified in much the same way, but with primers 5 and 8 rather than primers 1 and 4. These PCR products were separated by electrophoresis on an agarose gel and purified using the QIA Quick Gel Extraction Kit. After the enzymes and their dimers were amplified by overlap PCR, E. coli cells were transformed using plasmids and cultured. The cells were induced to express the proteins by the addition of 1 millimolar IPTG, a reagent that triggers transcription. The cells were harvested using centrifugation and stored overnight at -20 degrees Celsius. The pellets were then resuspended in lysis buffer the next day in the presence of lysozyme, DNase, 2X protease inhibitor cocktail, and sonicated. The lysates were collected by centrifugation. The insoluble lysis pellet that contained Fab-Z was solubilized. The lysates ran through an equilibrated and washed nickel sepharose column. The elution fractions were then analyzed for purity using SDS-Page, and yields were calculated by quantification and reported in milligrams per liter of culture. Fab-Z and Fab-Z dimer residues were calculated using the Scopes method since they had such a low abundance of aromatic residues.
The overall protein yields for Fab-A and the dimerized Fab-A were 40 milligrams per liter and 52 milligrams per liter of culture, respectively. However, the yield of Fab-Z was found to be substantially lower due to its highly insoluble characteristic. Only half a milligram per liter of culture of Fab-Z was obtained from the soluble lysis supernatant. On the other hand, the fused dimer Fab-Z was recovered in the soluble lysis supernatant in higher yields than the Fab-Z parent, 2.8 milligrams per liter of culture respectively. This helps to indicate that the dimer fusion helps to promote enzyme solubility. Our figure here shows the Fab-Z parent on the left and the dimerized Fab-Z on the right. The left indicates that more Fab-Z was found in the insoluble lysis pellet as compared to the soluble lysis supernatant. The dimerized Fab-Z shows that more dimerized Fab-Z was found in the soluble lysis supernatant than the insoluble lysis pellet.
Next, the researchers wanted to measure the enzyme activity, so they performed enzyme activity assays. And the enzyme activity was measured using crotonyl CoA as a substrate, since that reaction favors enol hydration, and we are testing dehydratase enzymes, so they will perform the opposite reaction. These reactions were followed spectrophotometrically by taking absorbance measurements over time to determine the amounts of crotonyl CoA and beta-hydroxybutyryl CoA in the mixture. Values for the absorbance slopes were calculated using an equation of micromoles per minute equals slope times reaction volume divided by path length times the molar extinction coefficient. The values were normalized by dividing the initial velocity values by total amount of enzyme and plotted as a function of substrate concentration to generate a Michaelis-Menten saturation curves. The values were then divided by Km values to get Kcat over Km, or enzyme efficiency values.
Fab-A in the dimerized Fab-A showed little to no activity towards crotonyl-CoA because it is shorter than 10 carbons long. Fab-Z only showed modest activity towards crotonyl-A, having a Kcat value of 144 seconds inverse and a Km value of 64 micromolars. Fab-Z showed a significant increase in activity with a 9-fold enhancement in catalytic efficiency, having a Kcat value of 3,814 seconds inverse and a Km value of 179.8 micromolars. These values can be depicted by this graph here.
The authors next performed size exclusion chromatography using a SuperDex 200 increase 10 over 300 GL column. Each run through the column was preceded by a run with standard protein mixture to generate a Kav versus log MW curve. The resulting standard curve was used to estimate the molecular weight of the proteins in the following elution fractions.
Using size exclusion chromatography, the quaternary structure for the different fused dimers was determined. The elution volume of dimer Fab-A corresponds to that of a protein weighing 45 kilodaltons. This elution volume is the theoretical molecular weight for the fused dimer and is in agreement with the dimeric nature of Fab-A. The dimerized Fab-Z has an elution value of 116 kilodaltons, indicating a trimer of fused dimers. This is consistent with the three-dimensional structure of Fab-Z, which has a hexameric arrangement. This hexameric arrangement is possible due to the fact that there is a formation of a trimer of dimers. In this figure, you can see that the elution volume of the dimerized Fab-A is located on the top, and the elution volume of the dimerized Fab-Z is located on the bottom.
In conclusion, we'd like to speak about some of the findings and applications of this experiment, as well as speak to some future experiments that could be done in studying fused dimerization to increase enzyme efficiency. Past strategies for enhancing fatty acid production in microbial cultures have included targeted deletion of beta-oxidation enzymes to cause fatty acid accumulation, the overexpression of thioesterases to increase fatty acid release, and the overexpression of transcriptional regulators governing fatty acid biosynthesis. This study proposed a complementary strategy to optimize enzyme efficiency by forcing dimerization through covalent linkage of dehydratase enzymes Fab-A and Fab-Z. As you can see in this figure, Fab-A and Fab-Z both catalyze the dehydration of fatty acyl ACP intermediates. Fab-A also catalyzes an additional double bond isomerization step to form the 3,4-cis double bond. These are both important reactions in the synthesis of fatty acids. The authors of this article showed many beneficial effects of fused dimerization of enzymes. These include increased activity, increased solubility, and increased expression. As can be seen in this graph, and as stated earlier, the activity of the Fab-Z dimer is 9-fold that of the Fab-Z parent enzyme. The Fab-A dimer, however, did not show much of a change in activity as compared to the Fab-A parent enzyme because the intermediate used in this experiment, carotenoyl CoA, is much shorter than the natural 10-carbon intermediate that's acted upon by Fab A. The solubility of the Fab Z dimer is much greater than that of the Fab Z parent enzyme, which can be seen in the results shown earlier, that the Fab Z parent enzyme was much more present in the insoluble pellet, and the Fab Z dimer was much more prevalent in the soluble supernatant. The researchers chose to use a rigid, naturally occurring peptide linker from a multi-domain polyunsaturated fatty acid synthase. The linker was rigid due to repeating alanine residues in its sequence, as evidenced by this magenta line, and the linker can be seen as this magenta connector in the figure. The linker, however, was still flexible enough to allow the dehydratase homodimers to arrange themselves in their natural forms. The Fab A dimer took on an intramolecular dimeric structure similar to that which Fab A exhibits in nature. The Fab Z dimer formed the expected trimer of dimers, and these were both evidenced by the size exclusion chromatography results. The results of this study suggest that fused dimerization has the ability to enhance the activity of naturally dimeric enzymes relevant to the fatty acid biosynthesis pathway. This could become an important tool for increasing commercial fatty acid synthesis in microbial cultures. Future work should be done to determine how these fused enzymes function within the context of their metabolic networks and whether overall efficiency and yields are increased.